Hello students, I am teacher Svetlana and in our today's lecture we will learn about the role of microbes as biofertilizers, which is covered in the chapter Enhancement of Food Production. So let's get started. Students, to understand the role of microbes as biofertilizers, we need to understand what are fertilizers. Fertilizers are nutrients which are necessary for the growth of plants and thus for the productivity of the cultivated plant. Use of fertilizers for increasing productivity is one of the aspects of green revolution. Fertilizers are classified as inorganic, that is chemical, and organic, which are biological. Inorganic fertilizers are synthetic, where mineral salts of nitrogen, phosphorus, potassium are mixed in definite proportions and then they are dusted in the field. Non-judicious or excessive use of such fertilizers lead to pollution of the soil, air and groundwater. Soil becomes acidic. Organic fertilizers are biological in origin and include farmyard manure (FYM), compost and green manure. Use of these fertilizers increases the fertility of soil. Nowadays, for better and sustainable agricultural production, farmers use biofertilizers and practice organic farming. Biofertilizers are mostly nitrogen fixing, living microorganisms which enrich the nutrient quantity of the soil. They include bacteria, cyanobacteria, and fungi. Biofertilizers are commercial preparations of ready-to-use live bacterial or fungal formulations. Their application to plant, soil, or composting pits helps to enrich the soil fertility due to their biological activity. Use of biofertilizers is cost-effective and eco-friendly. They play a vital role in maintaining a long-term soil fertility and sustainability. If you have a look at this image here, it illustrates the role of biofertilizers in helping improve the fertility of the soil. Now students, let us have a look at the type of biofertilizers. On the basis of the nature and function, biofertilizers are divided into the following groups. Nitrogen-fixing biofertilizers. The nitrogen-fixing microorganisms which convert atmospheric nitrogen into nitrogenous compounds like nitrates and nitrites which have ammonia. Nitrogen-fixing microorganisms are also called diazotrophs. They are of two types. Symbiotic nitrogen-fixing microorganisms, example Rhizobium, Anabaena, Frankia, they are always associated, that is, these are always present with the underground parts, that is, roots of higher plants. Then we have the free-living or the non-symbiotic nitrogen-fixing microorganisms. They are present freely in the soil. Example, Azotobacter, Nostoc, Clostridium, Bergeringia, Klebsiella, and many more. Then students, we have phosphate solubilizing biofertilizers. These are the bacterial species which solubilize the insoluble inorganic phosphate compounds such as rock phosphate. Example, Pseudomonas stratia, Bacillus polymyx, Agrobacterium, Micrococcus, Aspergillus species, etc. Compost making biofertilizers. Composting is a natural process that turns organic matter into a dark, rich substance called as compost or humus. The composting process is dependent on microorganisms to break down the organic matter into compost. There are many types of microorganisms that are found in active compost such as bacteria, fungi, actinobacteria, protozoa, and rotifers. Cyanobacteria that are used as biofertilizers. Many cyanobacteria are aquatic and terrestrial, free-living or symbiotic, aerobic, photosynthetic, nitrogen-fixing, heterocyteous or non-heterocyteous forms. Heterocyteous means they have a special cell where the process of nitrogen fixation takes place in their body. Now you have example Anabina, nostoc, Platonema, oscillatoria, etc. Anabina, nostoc, and Tolipotrix are associated with lichens, while Anabina is associated with plants like Azolla and Cycas. Then students, the next important type of biofertilizers are the fungal biofertilizers. Mycorrhiza is a fungus. It forms symbiotic association with the underground part like the rhizomes and root of the higher plants occurring in thick, humid forests. These are discovered by Frank in 1985. There are two types of fungal biofertilizers. They are called as ectomycorrhizae and endomycorrhizae. Let us have a look and try to understand what is ectomycorrhizae. Here is the image of ectomycorrhizae. They have well-developed mycelium. Mycelium is the body of your fungus that forms a mantle on the outside of the root. So here is the image of the root, and you can see outside a fungal sheath is formed. This is the mantle or the covering that it forms from outside the root. This increases absorptive surface area of the root and accelerates uptake of water and nutrients. These nutrients include nitrogen, phosphorus, calcium, potassium. Due to this, the plant's vigor, growth, and yield increases. Some hyphae of mycorrhizal fungus penetrates into the root and forms hearting nets in the intracellular spaces of the root cortex. Here you will see that the fungus penetrates and forms the hearting net structure, which is present in the intracellular spaces of the root cortex. So here in the intracellular space, it forms the hearting net. Now they does help to increase the surface area of root. More the surface area, obviously the amount of water absorbed will increase. Let us have a look at the second type of the fungal biofertilizer, which is called as the endomycorrhizae. Here is the image of your endomycorrhizae. They grow in between and within the cortical cells of the roots. Fungal hyphae penetrates the cells and forms finely branched arbuscules intracellularly and forms vesicles mostly in the intracellular space of the cortical cells. Here, have a look at this image here. It penetrates inside into the cortical cell and forms these arbuscules. Here, these arbuscules are formed, and hence they are also called as vesiculo-arbuscular mycorrhizae or VAMP. VAM. Nowadays, they are described as AM fungi. The plants with VAMP grow luxuriantly in less irrigated lands. The association of VAM with crop plants helps in conversion of less productive field into a more productive field. Now students, let us have a look at the biofertilizer microorganisms. The first one is Rhizobium. Rhizobia are rod-shaped, motile, aerobic, gram-negative, non-spore-forming, nitrogen-fixing bacteria containing the NOD gene and the NIF genes. Here, have a look at this image here. This is how the Rhizobium appears. They form symbiotic association with roots of leguminous plants. So here you can see in the roots of the leguminous plants, they form a symbiotic association. They bring about nodule formation on the roots and multiply inside the nodule. So they form these nodules and they multiply inside. They fix the atmospheric nitrogen into organic forms which can be used by the plants as nutrients. So they fix the nitrogen for the plants, and the plants in return supply carbon dioxide to the bacteria. Example, you have Rhizobium leguminosarum, which is specific to pea, and Rhizobium fascioli to beans. Here, have a look at this image. You can see this tiny round shaped nodules that are formed. These nodules are the place within which this bacteria Rhizobium grows and multiplies. Azotobacter, it is the important and well-known free-living nitrogen-fixing aerobic non-photosynthetic non-nodule forming bacterium intimately associated with roots of grasses and certain plants. It is used as a biofertilizer for all non-leguminous plants, especially rice, cotton, vegetables, etc. Here, have a look at this image. This is how Azotobacter appears. Furthermore, we have Azospirillum. It is free-living aerobic nitrogen-fixing bacteria associated with roots of corn, wheat, and jawar. It fixes a considerably large quantities of nitrogen in leguminous plants such as cereals, millets, cotton, oilseeds, etc. Here, this is the image of Azospirillum. Then we have Anabina. It is a genus of multicellular filamentous cyanobacteria that exists as plankton. It has abilities to fix nitrogen and also form symbiotic relationships with certain plants, such as the colloid roots of cycas and Anthracis thallus. It has some specialized and colorless cells called heterocytes, which are the sites for nitrogen fixation. Here, please have a look at this image. This is the Anabina cells, and you will see that it has different cells in it. Here you can see a small heterocyte. This is a special cellular region which is a site for nitrogen fixation. Then we have Azolla. Azolla is a free-floating water fern. Have a look at this image here. You can see they are just lifted up because they are freely floating on the surface of water. Azolla plant consists of floating rhizomes, that is stem. These are those floating rhizomes here with small overlapping bilobed leaves and roots. In the dorsal lobe, Anabina filaments are present in the parenchyma which fix the nitrogen. The leaf shows dorsal and ventral lobe. So this is the leaf of Azolla, and here you can see there that the dorsal side of your leaf there is a leaf cavity that is formed, and we have Anabina is present here which fixes nitrogen. The leaf shows both the dorsal and ventral lobe. Azolla can be used as biofertilizers in rice fields. Students, now let us see what are the benefits of these biofertilizers. Low cost, and they can be used by marginal farmers, free from pollution and the hazards which are associated with soil and water pollution. It increases soil fertility. BGA as biofertilizers secrete growth promoting substances, organic acids, proteins, and vitamins. Azotobacter supplies nitrogen and antibiotics in the soil. Biofertilizers increase the physiochemical properties of soil like texture, structure, pH, water holding capacity of the soil by providing nutrients and organic matter. So nowadays in our country, many biofertilizers are available in the market to reduce the use of chemical fertilizers and thus reduce the pollution. Thank you, students, for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe, and keep learning.